In this video, we will discuss whether dasatinib can play a role towards suppression of migrations of these pancreatic cancer cells and the mechanisms behind the suppression of these pancreatic cancer cells if our first hypothesis is true. So first what I did is they check whether this dasatinib can affect the migrations or not using the wound area healing method. So here what I did is they keep the cells in three conditions. First one is the untreated, and next other one is this 100 nanomolar of dasatinib, and the last one is this 200 nanomolar dasatinib. And what they are able to find out is that percentage of wound is completely recovered, or you can say the highest migrations is found in the untreated one, and with the 100 nanomolar they are able to see around 80% of the wound area, indicating that the greater suppression of these migrations. And with this 200 nanomolar they see a slightly more wound area, and similar kind of trends they have seen in the all the three cell lines that they have developed. And so in this 82739 cell lines they can see the around 80% of the wound area is still there, and for this 86119 they are able to see the 50% of this area is still here. So since they are able to see the suppression of the migrations, now they wanted to check whether how the suppression of migrations occurs. So what they did from their previous knowledge, uh, they have from the literatures they have the information that the dasatinib will suppress the SRC or suppress or affect the SRCC pathways. So they wanted to check whether it is because of this SRCC pathways that is causing the reductions in the migrations, or there are other factors also. So in order to validate this hypothesis, what they did is first they inhibit this SRCC using the this silencing RNAs that target SRCC, and they are able to see that at around 72 hours of this continuous treatment with this silencing RNAs, they are able to see suppression of the SRCC. So this is the condition that they have decided to check the migrations assay or the wound heal area, and in that case they are able to see that when they use this siRNA or when they silence this SIRC, when they silence the SRCC, they are able to see increase in the wound area or the reductions in the migrations. And these things they have observed for all the three cell lines, and the results are similar, or the trends are similar to the panel A, which is with the dasatinib treatment, and panel C is without dasatinib treatment. So this indicates that it's the siRNAs or the SRCC's pathways that plays a role towards these migrations, and the disruption in this SRCC uh, might affect these migration properties of the PDAC. Thereafter, what they did is they check these invasions in the untreated conditions and in the conditions where they use dasatinib. And what they are able to see is that when they use dasatinib, the invasions goes down to around from invasion goes down by around 60%, that can be seen here, compared to the control case where dasatinib is not used. And then they check the other pathways that are also get affected by these SRCC phosphorylations, such as FUC phosphorylations at Y861 and P130 phosphorylations, which is kind of similar to this SRC phosphorylations. And they can see that these pathways will also goes down with the expression of this pathway, this um, activations of this FUC and P130 also goes down with dose dependent increase in the dasatinib, that can be seen in the Western blot for both the activated pathways, that is FAK phosphorylations and P130 phosphorylations. And thereafter they check the literatures and they are able to see that this pathways, it is SRCC and FOC pathways, will help in the invasions in coordination with the MMP, in coordination with this MMP proteins. So then they check the effect of the dasatinib on the MMP proteins, and they are able to see that they are able to see reductions in this MMP9 and MMP2 when they are treated with dasatinib, and the results here are shown for no treatment and the treatment with dasatinib at 100 nanomolar. This in together indicates that dasatinib will suppress the SRCC pathway mainly, that results in the reductions in the migrations properties of the PDAC. And these results they have shown here in the in vitro conditions, and in the next video we will discuss their in vivo results also. So I hope you understood how this dasatinib and SRCC plays a role towards migrations of the PDAC, how this SRCC plays a role towards migration of the PDAC, and how the dasatinib and suppress the SRCC that results in the reductions in the migrations properties of this PDAC. Or in other terms, the SRCC inhibitors can act as a antimetastatic agents, and these things are validated in the in vitro studies. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.